My name's Steve Gallinger. I'm actually a surgical oncologist from Toronto. And I want to thank Jennifer and the organizers for inviting me. This is my first Cholangiocarcinoma Foundation meeting. And as a surgeon, I do certainly see HPV surgeon and see a lot of patients. It's a very impressive meeting, and I must admit that I wasn't aware of the magnitude and scope, so I'd really congratulate the foundation for the traction that they've obviously achieved and maintained over many years. It's not easy for advocacy groups to get a voice in the field. I've worked closely with colorectal cancer and pancreas cancer advocacy groups in Canada, and I just came from the Stand Up to Cancer meeting, which is a very impressive organization. So again, I would congratulate you on this meeting. And this is a topic that's very near and dear to my heart, so I'm really happy to chair the session on genetics of cholangiocarcinoma. In particular, I've also worked in colorectal cancer, and we do a lot in pancreas cancer genetics. And for those who are at ASCO GI, they may have heard Jennifer talk about our COMPASS trial, which is a high-content profiling program. And the concept or the importance of inherited genetics is just becoming obvious to those who it wasn't obvious to before. In fact, we showed in a lab that I've been running for a long time in Toronto in the mid-90s that germline BRCA mutations are not unimportant in pancreas cancer, but it's very hard to get the message across. The breast and ovary cancer groups, where there's lots and lots of survivors, have been talking about a germline predisposition to cancer, breast and ovary cancer and prostate cancer since the genes were discovered in the early 90s. But it's pretty apparent now that in pancreas cancer, at least 5 or 10% of cases are due to inherited predisposition, and I'm excited to hear what the speakers are going to talk about today. We've seen certainly cases of cholangiocarcinoma that are clearly caused by mutations in genes that many physicians just aren't aware of. So there really is a knowledge translation gap, and I think often the caregivers and advocates know more than physicians do about this particular field. So having said all that, we do have extra time today. There won't be the green, yellow, and red stoplight for the speakers because our middle speaker unfortunately had a family health emergency, so he's not coming, but it gives a longer time for the other two speakers. So I'm pleased to introduce Tony, who's known to his friends and colleagues, Bekaii-Saab, who's a professor at Mayo and has written much on this topic and will be speaking about the DDA damage response pathway in biliary tract cancer.
Thank you. Thank you for the invitation. Really happy to be here. I'm also glad not to have those blinking lights in front of me. I get confused very easily, so that will spare me that part. So I actually, was privy of the fact that I'm going to have a little bit more time. So Jennifer actually asked me to cover a little bit of both topics, which I think are quite interrelated. So I think we'll probably be able to cover a good 20-25 minutes on that and allow more time for the case and discussions. So I'm going to focus on the subject of targeting the DNA damage response pathway in biliary tract cancer. And I think that's a target that has become not just important in biliary cancer, but across the board. It ends up being, once identified, an Achilles heel for a number of malignancies, specifically in biliary tract cancer, as I'll show you. It's actually present at very high levels and therefore is prime for targeting. So as a background, if we think about our DNA, so every day there's thousands of DNA damage that occurs in our cells. And there are multiple different repair mechanisms to deal with them. So when that's happening, there's factories happening, you break a DNA, you fix it, break a DNA, fix it, and that's happening thousands and thousands of times a day. So BRCA, which is the one we know the most about, seems to interact with a number of other DNA repair proteins to form a complex system for DNA damage response, or DDR, and that includes all these other things that we call BRCA-like, BRCA-ness, or the likes. So this response system is a collective term. when you see the term DDR, that's essentially the collective term that points to these intracellular activities that are all linked to repair, to sensing and repair. So you sense and repair, you sense and repair. The response to DNA damage is different depending also on the cell cycle status. Just to make this more interesting is that, again, some of these proteins, although they're important in their function, they also depend on where in the cell cycle the cell is. So as such, DDR overall, as a blanket statement, is a very attractive source of drug targets, and I call it the Achilles heel, that can be exploited with novel cancer therapies. All right. So as a background, when we look at the TCGA, there's up to 193 DNA repair genes that have been identified, including 122 direct DNA repair and then 71 caretaker genes. 20 of the most frequently mutated DNA repair genes are listed here, including, of course, BRCA, ATR, ATM, FANK-A, FANK-D, POL-D, PAL-B2, etc. All these things we know. There are actually way more, as we said, than those, but these are the a little bit more easy identifiable. There are also the caretaker genes, which are also part of the response. They induce genomic instability, and that's BAP1, CDK12, and others. And again, these are important to keep in mind as we develop these targeting strategies for the future. When we look at studies with DNA repair genomic alterations, overall in biliary cancers and intrahepatic cholangiocarcinoma, they've been up to 12%, 26%, so the highest percent of those is actually in the extrahepatic cholangios, and then 8% in gallbladder cancers. So there are 6 pathways, at least, but these are the 6 major pathways to essentially repair DNA damage. And although these seem like they're separate pathways, they're not. They're actually interlinked and interact, but these are the major pathways, and you can see whether it's single-stranded, double-stranded. Single-stranded breaks essentially will lead to pressure of the cell, lead to essentially double-stranded DNA breaks, and ultimately get repaired through the HRD or occasionally through non-HRD. The most common is base excision repair, which leads to, which essentially is part of the single-stranded DNA break, and that's the one essentially that is prime for targeting with PARP inhibitors in the right setting. And we'll go through some of the understanding, because it's not really straightforward. You can just, you see a break, you block it, and it's going to produce its magic. It doesn't work like that. There are a lot of elements that work together to essentially make this somewhat challenging, but also creates a lot of opportunities for drug targeting, especially that we have a lot of these agents today available in the clinic, at least in clinical trials, but some even in the clinic. So this is where those PARP inhibitors seem to work best at that level of base excision repair. So I think since a lot of the focus in this whole area has started with the understanding around PARP and PARP inhibition, which is essentially poly-ADP ribose polymerase. Let's try to understand how PARP gets involved in repair DNA damage. Think about DNA damage, whether it's endogenous through cytotoxic therapy or radiation therapy. So how does this cancer cell ultimately repair its DNA when you damage it with alkylating agents, say, or radiation therapy? So you get the PARP polymerase, essentially, that binds directly to the single-strand breaks. So essentially, that's the sensor, that's the signal. And once bound to damage DNA, PARP modifies itself. It becomes that bridge, producing large branched chains of essentially poly-ADP ribose. So it's that bridge. And then this invites the repair enzymes to come in, and they stitch essentially the DNA, so repair the DNA, and then the DNA is repaired, and that's how cancer cells or cells in general deal with damage. So that's an important background to know as we move forward with this. So this whole concept of synthetic lethality, according to bracket status, is important to understand in that context. Again, it's not a simple equation of just using a PARP inhibitor and then killing the cancer cell. You have to have multiple elements, 2 at least we know of, more we're learning about. If you look at your, let's see, I'm a little bit dyslexic, but I'm on right and left. If you look at your left. The functional BRCA, sorry. That's why I need a Tesla or something that drives itself. I don't have a Tesla. I don't have a car that drives itself, but I should. But anyway, so the DNA damaging, whatever it is, that induces a single break produces essentially two elements that we know of. So imagine you have PARP. PARP is one of your first centers, and let's say you even inhibit that PARP. But you will have, with the pressure of the single-stranded DNA, you'll have this persistent accumulation of single strand breaks, which eventually will lead to double-strand break and collapse of the replication fork. That essentially triggers homologous recombination repair, which is essentially a function of a BRCA-competent cell. So that essentially, even in the presence, even if you block that PARP, then you use alternative pathways as well through patch repair, and then again through the homologous recombination. And that's in the presence of the dysfunctional BRCA, where you don't have that capacity to essentially do the repair. This is how PARP inhibitors will induce their maximal effect. Now, keep in mind that even if we look at the most sensitive cancers, the most sensitive to BRCA, to PARP inhibition, even BRCA mutated, 30 to 40% response rate, which essentially means that this is one mechanism, and without going through excruciating details of the other mechanisms, because we don't understand them too well yet, there are a lot of other mechanisms that we still need to understand about why we see some resistance in some of these patients, but that also is an opportunity for us to continue to advance science. All right, so this is it's somewhat of a simplified way to think about cell death from synthetic lethality as induced by PARP. So starting from A all the way to D. So think about your normal cell. And you have base excision repair primarily through PARylation and PARP1, homologous recombination repair primarily through BRCA. So that's how essentially DNA gets repaired. Now, think about your cells with the BRCA mutations. Now, we know that cells with BRCA mutations have an increased propensity essentially to certain cancers, including, of course, cholangiocarcinoma and biliary tract cancers. Homologous recombination pathway will be affected, and you'll be left with a base, essentially with the base excision repair through PARP1 for repair. So you still find a way to repair that DNA. Now think about your cells with drug-induced PARP inhibition, whether it's a PARP inhibitor or other or other agents like platinums or others that could affect that pathway. And you affect essentially the base excision repair. In the presence of an intact BRCA and homologous recombination, essentially the DNA will still find its way to repair itself. Now, so the best setting, of course, for these agents is cells with BRCA mutations and then PARP1 inhibition. And those are the ones where you can essentially induce a break and then affect repair and then kill the cancer cell. But it gets more complex than that. I wish it was a simple story. It's not. But we understand more of it, and that's important. So the DDR pathway targets are many, and we talked about 6, but there are probably more than that as well. And there's a lot of different ways to break DNA and repair it. The most common lesion is actually a single strand break. The repair targets are typically PARP and another one called AP1, repair pathways through base excision repair. And the damaging agents here are primary radiation therapy, alkylating agents, or a mutation. So this is the most common lesion, and that's why this is the one that's being studied the most and first. Then you have the double-stranded strand breaks, ATR, ATM, and others, and we know that there are agents that are being developed, or chemotherapeutic agents that specifically, like topoisomerases, that can affect ATM, and then other effects as well. So this whole homologous recombination pathway, or repair pathway, as we call it, is essentially how PARP interacts with all these proteins, part of the DNA damage response, repair response. And you can see the complexities of this. When you essentially induce DNA damage, there's increased expression of all these genes involved in DNA repair. whether it's ATM, ATR, or others, BRCA. And so all these are working together. If you can think about this as a factory, all these things are working together to sense the problem, repair the problem. If, one man is down, another one picks up and helps with the repair. So this is really how it works around this pathway. And that's, again, why we don't see a one-to-one response with PARP inhibitors, even in the best setting, or even with combinations. So what do we know about HRD prevalence? And of course, we want to focus on biliary tract cancer. And this is actually now in press in JCO Precision Oncology. This is from the CARES database on 52,000 solid tumors, including 870 biliary tract cancers. HRD was defined as we typically define it in the pathogenic mutations of all these things listed. So this was a molecular profiling study with next sequence selected 592 genes. So pretty standard. Interestingly, what was found is if you look by lineage for HRD mutation frequency, biliary tract cancer was second to endometrial. So very high level of HRD mutation frequency, close to 28 to 30%. Now, interestingly, if you look at all the cancers combined, it's about 17%. So that tells us that this is a very, very important aspect of carcinogenesis, but also since we know this is targetable, at least parts of it are targetable, and we're learning more, that also makes it a very interesting target to understand, since it's so prevalent across multiple cancers, specifically here biliary cancer at a high level, and then we have agents in one way or the other that can target this, either singly or collaboratively. And this just shows, again, the HRD mutation landscape. Again, you can look at the difference. ARID1A is one of the primary drivers, but this just lists all the elements with biliary cancer. That's your red arrow. All right, so there's a phase 2 study that's sponsored by ICRN and supported by AstraZeneca that's going through our research network, a crew that's looking specifically at the role of PARP inhibition with olaparib in patients with advanced biliary tract cancer expressing aberrant DNA repair, based on the fact we expect about 20 to 30% of these patients to have those. We know about some cases, and we'll hear a little bit about that, some cases that, in the presence of BRCA mutations have had great responses. We don't know much about the HRD tumors. We know from other cancers that HRD-driven tumors, may respond to these agents, perhaps not as well as BRCA mutations, but there is some element of response. And so that study is exploring specifically that question. So going back to, strategies about how to best combine and understand how we stack now our decks in the right way, stack our cards, so we can essentially induce the maximal benefit from combining these agents together. So you think about the cell cycle, that cell goes through different cycles from growth phase, replication of DNA, then you get growth phase 2, G2, and then you get the cell to divide. That's normal, but that's also what cancer cells do. They go through the cycle, breakdown, multiply, breakdown, multiply, breakdown, multiply. So when you think about that process, you know that in the process, DNA is replicating, and DNA is your target. If you can, break that DNA, you're going to kill that cancer cell, and you're going to stop it from dividing and essentially multiplying. So two things when you think cooperatively about how to do it, because different parts of the cell cycle seem to be more sensitive to different manipulations. So on the G1S interphase, you want to maximize your damage. Now, when you maximize damage, just remember that cell is going to go, or the DNA is going to go into an attempt for repair mode to salvage the cell and essentially go to the M phase where it multiplies and essentially, continues to multiply and form tumors. So if you can maximize your damage, and that's typically what certain cytotoxic agents, alkylating agents, or or radiation or topoisomerase inhibitors like irinotecan would do. And then on the other hand, you prevent repair, and that's essentially what PARP inhibitors could do. Although PARP inhibitors can be also poisonous to the cell, but there's also ATR inhibitors, check inhibitors, and others that could essentially be in that world. Now, and then ultimately, you know, the effects of all this will manifest themselves in the end phase, and this is the cell prevented from multiplying and then eventually dies. And that's ultimately what we'd like to achieve with thinking about rational combinations and where to place them and how to sequence them and how to work, you know, chemotherapy plus PARP inhibitors, strategies that have been challenging. We'll talk a little bit about this. But then now we have this whole angle about understanding how all these different, the genomic instability and these damages essentially lead to increased tumor immune-mediated cell cytotoxicity, and how we can take advantage of a lot of these changes and think about introducing immune therapy along with these PARP inhibitors or other DDR inhibitors, excuse me, in biliary cancer. So talking about combining these DDR agents now, thinking about primarily two elements, but there are more that we can think about. So you can think about multiple permutations, and some of them are actually, you know, being looked at in clinical trials, and others in much earlier phases, and others, you know, being postulated. But the two that are actually making the most segue are how to combine these DDR agents with chemotherapy, and not any chemotherapy, specifically alkylating agents and topoisomerase inhibitors. Two, how to combine these agents with immune therapies, and that's number two, and that's being built up. So if we think about the homologous recombination, specifically BRCA, but also others such as ATM and ATR, and how they predict for response to certain anti-cancer therapies, such as platinums and topoisomerase inhibitors, and we know that there's a lot of literature that supports that and a lot of rationale to do that. So these two prime agents prime for development in combination are the topoisomerase inhibitors, specifically irinotecan, but also agents like temozolomide and platinum salts such as oxaliplatin, cisplatin, and carboplatin. And a lot of that is already, or has already been looked at in cancers like breast and ovarian, and being looked at in pancreas, and trickling its way down also to biliary tract cancer. The problem and the biggest challenge with these agents are their overlapping toxicity, specifically diarrhea, GI toxicities can be quite tough, quite horrendous if you put them together. And multiple studies have actually looked at the combination of the two. And many of them were discontinued because of toxicities, at least with irinotecan, which makes a lot of sense, actually more sense than the alkylating agents to combine, since there are two different pathways that they act on versus, you know, the alkylating agents and the PARP are kind of overlapping in terms of their targets. It never really made a lot of sense to me to combine a platinum with a PARP inhibitor as much as a topoisomerase and a PARP inhibitor, but that also was riddled with challenges of toxicities. When we think about all this, when we look at all the studies that essentially assessed topoisomerase inhibitors and PARP inhibitors and put them together, one theme always stands out is when you sequence them, you end up with the best tolerability, but you also start seeing some responses. And it kind of makes sense when you go back to the cell cycle, as we talked about it, you know, you want to maximize your damage and prevent repair. I mean, this is really how you should do it. We should sequencing. Because if you look at the continuum of cancer cells, they're not all synchronized. They don't all essentially cycle at the same time. They all cycle at different times. So your best hit is to essentially use your agents frequently, but also in sequence to be able to hit that cycles at multiple turns, multiple times. When we think about PARP inhibitors, going back to PARP inhibitors, because those are the most advanced in development, these exhibit their activity through two mechanisms. One is what we call a catalytic PARP inhibition, but the other one is PARP trapping, PARP. Now, we have a number of these PARP inhibitors on the market or in development. Veliparib is the one that's weakest, and that's actually probably never going to make it anywhere in the clinic, even the most sensitive cancers. Unfortunately, it's the one that's been studied the most and has been essentially painfully, in many ways, just omnipresent and limiting access to other agents. Thankfully, now we see, these rucaparib, olaparib, niraparib, talazoparib, all these, ribs. I'm sure there are more than listed here, that are making their way slowly but surely. These actually end up having more trapping activity. Veliparib does not have trapping activity. One of the things about veliparib, and the reason why it was so popular in drug development, is the thought was that it is the easiest one to combine with chemotherapy, and primarily with irinotecan. And the reason for that is essentially it doesn't have that trap activity, so you can use it at a lower dose, and it still has that catalytic activity that has its effect. So this was based on one preclinical study. Comes another preclinical study from actually Kaufman from Mayo, that suggested the exact opposite, that to synergize with topoisomerase inhibitors, you do actually need the PARP trapping activity. And without it, you do not synergize. So it goes against the other paper. Unfortunately, a lot of work has been done in the meanwhile clinically that certainly, put a little dent on that hypothesis. That said, knowing that it's important to do that, and knowing that there are overlapping toxicities, so how do we overcome these obstacles as we move forward? So we talked about sequencing strategies, but then there are issues of overlap, meaning that, and I'll show you that in the next slide, if you have your irinotecan going in, within 2 days, that's irinotecan is off your system, but this is where you should probably start your PARP inhibitor. So you're really not giving any chance for overlap, which certainly can cut down on the chances of synergism, but also that may be the only way feasible. So other ways to do them is actually to use formulations that selectively deliver active agents to the tumors. If you can just selectively, liposomal formulations, theoretically, there are other formulations that may deliver these chemotherapies directly, and so you essentially enrich the chemo in the tumor, and you minimize the risk of toxicity with accumulation in normal organs. So we actually have embarked on a study with Naliri 5-FU and rucaparib in pancreas and other GI cancers, but also will include a biliary cancer cohort. And that's based on the knowledge, and I'm not going to show you every work we've done but this is the gist of what exactly why exactly we thought this would make sense, and this was corroborated by more preclinical data than I'm showing you in this slide. But as I said, if you think about your irinotecan and your maximum exposure, it's 48 hours. Within 48 hours, most patients would have cleared irinotecan from their system. So when you want to try to find your peak overlap with the PARP inhibitor, you're almost under obligation to either start at the day you give the irinotecan or the day after. I mean, the later you do it, you're going to have almost no SN38 left in the tumor, and you miss essentially on that capacity to do that. So nanoliposomal irinotecan, and I understand, certainly that perhaps head-to-head there may be not much of a difference between the two. I'm not sure that they were compared head-to-head, including in a small study. But nanoliposomal irinotecan is actually particular in terms of its pharmacokinetics. So it stays in the system for about a week, at least a week, actually. And the interesting part about it is that in the first 48 hours, it almost is completely clear of the normal organs. I mean, these studies have been done. So meaning that within the first 2 days, the drug is clear from most of the system, like irinotecan. However, it persists in the tumor, at least SN38, which is the active component, for at least a week. So which makes it kind of interesting. And so we just started a phase 1 study with that sequencing approach, hoping that indeed, what was found essentially with the kinetics of the drug was some of the preclinical work that we've done and this rationale that you can essentially give the nanoliposomal irinotecan, and then you start the PARP inhibitor 3 to 4 days after, hoping that SN38 is still at high enough levels in the tumor, but has cleared most of the organs. So far I can say that studies has been going fairly well, but we're still in the early phases. So hopefully it will actually be able to show clinically that proof of concept. All right, so let's move to the next frontier. You know, there's no talk that is interesting without using the word immune therapy. I mean, I can talk to you about all the chemotherapy you want, about the exciting discoveries, but say one word about immune therapy and everyone's flashlights go on. It's exciting, but it's not for everyone. We have to know how to use it and where to use it, frankly, because it has its own toxicity, including its financial toxicity, but it's also a big element of toxicity. For someone, you start stacking them along, I mean, you end up with toxicity. But for the right patient, you get some significant and very meaningful responses, and that's really where we need to focus, you know, our thoughts. So we know that there's a lot of interaction between the two. These are tumors that have the capacity to induce a very strong immune modulating response. Because DNA damaging agents overall, when you damage the DNA, essentially lead to cell death, which is immunogenic by itself. You stimulate neoantigen production, and you know people can argue about what antigen and neoantigens mean. It's quality, quantity, whether it matters or not. But it's definitely, at least at the micro level, does tend to inflame things and alters the inflammatory milieu. So this essentially is one characterization, and you look at the hexagon, I guess, or what is this? Whatever, 6. The modes of action. Let me see. I mean, I don't know if I can. No, I can't. Anyways, if you look at what these DNA damaging agents would do, both at the tumor cell level, as we said, immunogenic cell response, they break it, they release the antigen. But I think more interestingly is what they do, what a lot of these agents do actually at the level of the milieu itself, the immune milieu, where they inhibit Tregs and MDSCs, which, as we know, are bad actors. These are the ones that essentially inhibit the good cells from doing their work. And so, you know, if you give these PD-1 inhibitors, in my simple mind, if you give these PD-1 inhibitors and CTLA-4 inhibitors and other checkpoint inhibitors, you know, to agents that essentially are to tumors that are incredibly immunosuppressive, I mean, you release the brakes on cells that are going to hit the wall. You know, they're not going to do anything. They keep on hitting the wall and just doing nothing. So if you can break the wall, essentially, by inhibiting the Tregs or the MDSCs, then if you release the brakes, you know, these cells are going to go and do action. And so that's the whole primary reason why, you know, the thought is that you could combine these modalities in a way that would actually be productive for immune therapy and for maximizing killer cell. I'm looking at Greg Lazinski over there to see if he's agreeing with anything I'm saying, or he thinks I have, like, you know, 2 heads coming out. He gave me the thumbs up. Thank you, Greg. All right, so now going back to this whole idea about the interaction between those genomic alterations and the tumor mutational burden, and potentially, you know, how can we essentially take advantage of the two to maximize benefit, essentially make PD-1 or inhibitors or other immune strategies work in diseases where we have very little activity, and how can we identify those with the highest potential for damage, for DNA damage, and prevent repair, and essentially make them more susceptible to these immune therapies. So one thing, and this is from Ham and Milland, this is looking at a cohort of Chinese versus US patients with intrahepatic cholangiocarcinoma. So it's important to see that, you know, again, when we think about cholangiocarcinoma, we've always suspected that there are differences, ethnic, regional, geographic differences. It makes sense. I mean, you know, this is a disease that interacts with its genetics, and its genetics interact with its environment. And the complexities of the environment are very different if you look at parts of the world. So you can see that in the Chinese cohort and the US cohort, there are definitely different drivers. There are some commonalities, but there are definitely different drivers. This is from a paper that is a collaborative paper between the groups, led again by Reham and Milland, and Mayo was part of that. But this study just, looking at the variations in DNA repair gene alterations and tumor mutational burden to try to understand, where do they intersect, and are those the right patients to pick ultimately for therapeutic manipulation. So, excuse me. Yeah. So we can see that the presence of tumor mutational burden high tumors in the direct is actually at low levels, 2%, 1%, both 7%. So this is not, in 1 to 2% of the patients will see these TMB high, which may explain why we don't see these magnificent responses in every patient with biliary cancer. When we look at intermediate, it's much more than that. And intermediate is actually the best opportunity probably to think about combinatorial agents. And that's actually at 18%, so that's not insignificant. And the low 80% are probably the ones that are most challenging. And again, looking at the differences between Asian and US-based population, there's definitely differences between the two, almost at double the rate in China versus US. So there may be some different drivers. Not insignificant in the US, but definitely more significant in China. So not going through all this, but this just to show you a lot of this work is being done, the PARP plus these inhibitors. And the thought is, depending on whether you have really a high tumor infiltrating lymphocyte burden or not, whether you start with a PD-1 inhibitor and then add the PARP or the DDR inhibitor, or start from the get-go in those TMB intermediate or low. All right, so I'm not going to go through this, but just to tell you that there is life beyond PARP, and so this is a lot of work that has and will be ongoing in this area. So as a conclusion, I think this is quite an attractive source of drug targets. And a lot of strategies are being looked at, including rational combinations with chemotherapy, emerging combination with immune therapies, understanding a little bit more mechanism of resistance to continue developing agents in this field. And I think at the end of the day, one thing we need to keep on reminding physicians and even patients, reminding their physicians that everyone should get genotyped for the tumor, routine gene testings whenever available, if possible. Thank you.